Okay, morning everybody. Before we start today's lesson, we're going to do a quick recap quiz on a previous topic. So on your screen now, you've got 6 questions from previous topics that you've covered. I'd like you to just pause the video and just write those questions down in your book and write down your answers. And when you've got all 6 answered, play the video again and we'll go through the answers together. Okay, so let's go through those answers. Question 1 was, what process happens in a mitochondrion? And the answer is aerobic respiration. Give yourself a tick if you got that right. If not, please correct it. Question 2 said, when you look down a microscope, what is the area that you see called? And the answer to that is the field of view. So if you got that, well done, give yourself a tick. If not, just correct that as well. Question 3 said, what do we call the change of state from a liquid to a gas? And when something's changing from a liquid to a gas, we call that evaporation. Well done if you got that right. Question 4 said, the melting point of sodium is 97.7 degrees Celsius. What is the state of sodium at 25 degrees? So 25 degrees is less than the melting point of sodium. So if it's less than the melting point, then sodium must be a solid at 25 degrees, and that is the correct answer. Question 5 said, what does a vector have that a scalar does not have? And vector quantities have a direction and a magnitude, and a scalar only has a magnitude. So the answer to that question is direction. Tick if you got that right. And the last one said, what is the equation for calculating speed? I'm sure you've all got this right. Speed is calculated using the equation speed equals distance divided by time. Well done if you got all of those correct. We're now going to carry on with today's lesson, which follows on from our previous lesson on cell division and mitosis. So you might want to put the heading in your book, or maybe you're using a Word document, put the heading Mitosis Part 2. So we talked yesterday about the cell cycle having 3 main phases. They were called interphase, mitosis, and cytokinesis. And we're just going to quickly just recap what each of those phases actually means. Interphase, the first stage of the cell cycle, is when the cell increases the number of subcellular structures, such as mitochondria and ribosomes, so that the cell can divide in 2 and each new daughter cell has enough of those structures to survive. The cell also replicates or copies each chromosome so that the 2 new daughter cells each have an exact copy of the original DNA. The second stage of the cell cycle was called mitosis. This is the stage when the chromosomes move apart and the nucleus divides. The genetic material which has been copied is now split in 2 and divided amongst each side of the nucleus. The third stage was called cytokinesis, and this is when the cytoplasm divides into 2, creating 2 new daughter cells, each now with their own copy of the original DNA. Whilst describing the cell cycle in these 3 different sections gives us a good overview of how the cell cycle works, unfortunately the second stage, mitosis, can actually be divided down into 4 more detailed sections. In this lesson, we need to take a closer look at each of those 4 stages of mitosis. So first of all, we're going to run through what each of these stages is called. We should, as a minimum, be able to remember the names of all of these stages and have them in the correct order. And these stages are called Interphase, which we've already met, Prophase, Metophase, Anaphase, Telophase, and Cytokinesis. So you can see that interphase and cytokinesis were in our original description of the cell cycle, and prophase, metaphase, anaphase, and telophase are the 4 stages of mitosis itself. Now, to make sure we can remember the correct phases in the correct order, we can use the mnemonic "I peed myself and then cried." So your first task today is to pause the video now and then write down in your book. You can put a heading Mitosis Part 2, and then you can put down the name of the 6 different phases of the cell cycle and the mnemonic as we've got on the screen now. So just pause the video now and do that, and then press play when you've done. Okay, so we're going to talk through what happens in each of the stages. In each of these stages, there are 1 or 2 really important things you need to remember in order that you can answer exam questions on this topic. So let's have a look at prophase first of all. So in prophase, the nucleus starts to break down and spindle fibers appear. During metaphase, the chromosomes are lined up on the spindle fibers across the middle of the cell. That's the only thing you need to remember for metaphase. The next stage is called anaphase, and during anaphase, the only thing you need to remember is that the chromosomes are pulled to either end of the cell by the spindle fibers. During telophase, a membrane forms around each set of chromosomes to form 2 nuclei. So we've gone from having 1 nucleus with 1 set of DNA to having 2 nuclei, each with their own exact copy of the original DNA, although we still technically have 1 cell because the cell membrane at this point is still intact. Our final phase is cytokinesis, and we've met this before. This is when the cell membrane separates and reforms around each nucleus, creating 2 separate daughter cells. What I'd like you to do now is pause the video, and in your book or on your Word document, I'd like you to either recreate the diagram that's on the screen now, or maybe just make a table if you are not able to draw that diagram out. And for each stage, obviously put the heading of each stage, but also put bullet points about what happens during that stage. You don't need to write the full sentence of everything that's on the screen. Just try and summarize it down into a bullet point. So for example, for anaphase, you could simply put the bullet point, spindle fibers pull the chromosomes to either end of cell. So pause the video now, complete your diagram and your bullet points, or your table with your bullet points, and then press play when you're done. Okay, let's see how you've got on. So on the screen now, we've got 4 questions. I'd like you to write these questions down in your book or on your Word document, and then write your answer after each question. Pause your video whilst you do that, and then we'll go through the answers together. Okay, so question A asked which part of the cell cycle is taking place when the chromosomes line up on the equator of the cell. If you look back at your diagram or your notes, hopefully you can see that that is metaphase. So give yourself a tick if you got metaphase. If you didn't, then correct that. Question B asked you which part of the cell cycle is taking place when the nucleus membrane is breaking down. Looking back at your diagram, hopefully you can see that that is prophase. That is the first stage of mitosis. That is when the nucleus breaks down and the spindle fibers first appear. Question C. Which part of the cell cycle is taking place when DNA and subcellular parts are copied? Now that won't be in your diagram because that is interphase, which is actually before prophase, so it's not actually on your diagram, but you should know that from our previous lesson. So interphase is the answer there. Please give yourself a tick or correct that. And the last one was D, which was which part of the cell cycle is taking place when the cytoplasm of the cell is separated. And that, hopefully you got, that is the very last thing that happens. We've got two nuclei, two copies of the DNA, and finally our cytoplasm separates and we form two new daughter cells. That's cytokinesis. Well done if you got that. Give yourself a tick. If not, give yourself a correction. So we've looked at how living organisms use the cell cycle and mitosis to produce new cells. And we've seen that this happens so that living organisms can repair tissue and grow. Normally, cells will only divide when they need to. However, sometimes the process of cell division can go wrong, and this sometimes leads cells to turn into cancer cells. Cancer cells undergo uncontrollable cell division. Rapid, uncontrollable cell division can produce lumps called tumours, which can damage the body and sometimes result in death. I'd like you just to watch the short video that's coming up that explains this process in a bit more detail.
Our bodies are made up of 100 million cells. They're so small you can fit 100 cells on the top of a pinhead. Most cells have a nucleus, cytoplasm, and cell membrane. The nucleus is the cell's control centre and holds the cell's DNA. Your DNA carries all the instructions needed to build your body and maintain its functions. Each instruction is carried on a unique piece of DNA called a gene. Cell growth and multiplication is part of a continual process called the cell cycle. To move through the different stages of the cell cycle, each cell has to go through a series of checkpoints. These checkpoints act a bit like traffic lights, and they only give the green light to a cell that's ready to go through to the next stage in the cycle. Only healthy cells are allowed to multiply, and so the cell cycle must control which cells are allowed through. The DNA inside cells can sometimes be damaged, either by mistakes in the normal processes in our cells or by things in our lifestyles or environment. Damaged cells are stopped at checkpoints and either repaired or destroyed. But occasionally damaged cells can find a way to sneak through. And sometimes the faulty DNA instructions may tell the cell to multiply at the wrong time or in the wrong place. If a cell accumulates too much damage over time, it can become a cancer cell. They then multiply out of control to form a lump or tumour. This is how cancer starts.
Okay, so the very last task I'd like you to complete today is to return to Go for Schools and click on the link and complete the exit quiz, which contains questions about today's lesson and what we've discussed today. If you have any questions at all, please email me and ask for help. Well done, and I will speak to you tomorrow.